Welcome back, students. So today's topic of discussion is some advanced methods of microbial preservation. Earlier, I have discussed with you all about some short-term and long-term methods of preservation. So please watch my video till the end and do subscribe my channel and do like my video. Why do we need to study some advanced techniques of microbial preservation? Because the conventional microbial preservation techniques have certain limitations and do not show promising result in terms of maintaining the viability and functionality of the microbiome during long-term preservation of intact samples. What do we understand by intact samples? Intact samples are those which we collect from any environmental sites and want to preserve them directly after the collection. So for that purpose, the first technique is cell alive system technique for intact microbiome preservation. This technique is originated from the Japan. This technique was developed for the food industry, especially for the preservation of raw seafood items. This technique is based on the freezing technique. The cask freezer technique is based on a very simple principle that the spinning motion of water during freezing prevent the formation of ice lattice or ice crystals. In this system, presence of alternating or oscillating magnetic field and mechanical vibration induce uniform cooling and minimize the size of crystal formation in the preserved sample. This process maintains the natural texture and prevents the cellular and molecular damage in preserved samples and further claim to influence the tissue survival rate. So it enhances the survival rate of microbiome as well as the tissue. What are the few advantages of cast technique? First, the spinning motion also helps in minimize the freezing point of water that assists longer freezing effect and reduce the time required to achieve the super cool status of water. Second, samples preserved in cask freezer showed more viability than sample preserved during other freezing technique. Cache freezing technology has been approved as well optimized tool for long-term preservation of the sample. Cache freezing technique to preserve subsea floor sediment samples for biomolecule-based microbial abundance studies and achieved a satisfactory result. So this technique has advantages, or we can say have advantage over other freezing technique because it can decrease the formation of ice crystal which can happen during the other freezing techniques. Second technique in advanced microbial preservation is gelatin disc method. As we all know, gelatin is a solidifying agent, so we can use it for microbial cell preservation. First of all, we have to understand why we need to use this type of method. Lyophilization is a widely accepted method for long-term preservation, storage of microbial samples, and for transportation purpose. But due to excess cost of instrumentation, many small laboratories and colleges cannot afford this method. There is no direct report on use of gelatin disc method for preservation of intact microbiome, but due to their capacity to maintain viability and functionality or functional stability of a number of bacterial strains, researchers might expect this technique as a possible alternative for long-term preservation of microbiome. So, bacterial survival ability after drying and simple bacterial preservation technique using gelatin disc. This method gives good result for successful preservation of several pathogens but did not perform well for prolonged preservation of Vibrio cholera and Neisseria species because these species are fastidious organisms, means they need some extra growth factors such as for Vibrio cholera they need salt and for Neisseria they need serum for the growth. By using this technique, some researchers can maintain beta-lactamase activity of Pelicinase producing and gonorrhea for 3 years, and they have also found that gelatin disc method allows survival of fastidious organisms up to 1 year at -20 degree storage. We can also preserve some thermophilic bacterial spores on filter paper. Spores of thermophile can be preserved at various temperature ranges from 4 to -20 degrees Celsius for almost 1.5 years. This is the protocol for gelatin disc method. First, we will place the bacterial suspension drop on gelatin on the sterile Petri plate. Then we will dry the drop over phosphorus pentoxide under vacuum. Phosphorus pentoxide is a white crystalline compound and it has a dehydrating property. So many bacterial species can be preserved using this method, which can be belong to Enterobacteriaceae family, Haemophilus, Neisseria, Flavobacterium, Streptococcus, Gymnella, Pseudomonas, Bacteroides, Granhamella, and these bacterial species can be preserved for up to 1 to 5 years using this method. Third technique in advanced microbial cell preservation is silica gel preservation. By this technique, we can also preserve some microorganism, or we can say fungal spore or spore producing microorganism up to 30 years without any changes or mutation in the microbial species. For as you can see, this is the protocol or the procedure how we can preserve our microbial cell using silica gel beads. This is the bacterial suspension. In this, silica gel is added with lyoprotectants. Lyoprotectants are those agents which can help in the preservation of the microbial cell in harsh environment or the freezing environment. They prevent or preserve these cells from the damage such as DMSO, glycerol, hyaluronic acid. Then we will dry the cell suspension and partially after that partially desiccated silica gel will form and these can be preserved for the long-term storage. Freeze drying lyophilization is a method of preserving bacteria and microorganism or other microorganism. It involves drying the microorganism on sterile silica gel granules. Silica gel is a porous form of silicon dioxide. It is made up of three-dimensional framework of alternating silicon and oxygen atoms. The framework has nanometer scale void and pores. These voids can contain water or other liquid or can be filled with the gas or vacuum. Microbes can be stored in silica gel powder at low temperature for 1 to 2 years. Species can remain viable for 10 to 30 years without undergoing change in their characteristics. Technique is cellular immobilization or entrapment. Formation of microdroplets or beads with different gelling agent like alginate or acacia gum is well-known method of microbial cell entrapment. Several factors like size, texture, porosity, surface to volume ratio, and chemical nature of gelling agent impact the cell viability and functionality during the preservation process. Several organisms of probiotic importance like Bifidobacterium and Enterococcus have preserved using this method and giving good response. Encapsulation followed by drying is a good option, but freeze drying did not give good response due to osmotic imbalance and oxidative stress created during the freeze drying process. That's why we do need some advanced technique of microbial cell preservation. Fluidized bed drying gives better response in terms of viability and functionality. Fluidized bag drying gives better response in terms of viability and functionality. Furthermore, addition of cryoprotectant and antioxidant in gel matrix provide long-term viability and functionality to entrapped cell. It is also noticed that use of biopolymer like acacia gum and pullulans is better than inorganic polymers. Technique is electrospinning and electrospaying. These are the two techniques through which we can preserve microbial cell for longer time. Electrospraying is also known as microencapsulation in microbiome preservation. Microencapsulation method of preservation work better for such type of microbes and is used to overcome such difficulties. High voltage electrohydrodynamics processes like electrospaying and electrospinning are giving good result in preservation of microbial cell. Electrospinning and electrospaying are novel microencapsulation techniques used for maintain the viability and functionality of microorganism. Generally, both technologies are known as sister technologies because they resemble with each other because they because the way of working of both technology are almost similar with only differences in viscosity of polymer solution. In electrospinning, polymer solution is high viscous, while the viscosity of polymer solution is in electrospinning is low, which gives nano or micro scale fiber of polymer entrapped microbial cells and smaller droplet speeds respectively. This is the instrument which we use for electro spring technique. As you can see, this is a syringe pump with polymer solution and microbial cell. This is the Taylor Kong with high voltage DC supply. This will push the polymer solution and microbial cell. This will create a weaves. This will form a weaves through which in which microbial cell is entrapped. These are the fiber and beads with entrapped bacterial cell. These are the fibers and these are the beads. These both contain microbial cell. So through these techniques, we can preserve different type of microorganism for longer time. And these techniques are better than the short term or long term method which I have discussed earlier. do like my video and please do subscribe my channel for more such informative video. Thank you.